So our next topic is going to be glycogen metabolism. And all glycogen metabolism is the conversion of glucose into glycogen or the conversion of glycogen into glucose. And the direction depends on the state of the cell. So if the cell has a lot of energy, it's going to go convert glucose to glycogen. If the cell does not have that much energy and it needs energy, it's going to convert glycogen back to glucose. And just a reminder of what these molecules are. We're pretty familiar now with glucose. We've seen it several times this semester. Glucose is a monosaccharide, and it's used as an energy source for most organisms. And we saw that when we saw the pathways of glycolysis, the citric acid cycle, oxidative phosphorylation. We know that we can convert glucose into energy, mainly ATP. And where can we get glucose? We can get glucose through our diet. That's the main source. But we can also get it through gluconeogenesis, which is a process we'll learn about next in a future lecture. Glycogen is a polymeric form of glucose. And when we studied carbohydrates back a while ago, we learned that glycogen is basically just chains of glucose molecules. More scientific way to say that is we have polymers of glucose. It's found mostly in the liver. There is some also in skeletal muscle as well. So that's the main source of or the main area where we find this molecule. So we really don't see glycogen in every single cell in abundance, but in the liver and muscle we see an awful lot of it. And this is our most immediate large-scale source of energy. So when we really need some energy at any given time, this is where the body goes to get glucose. And so if we look at a liver tissue here, you can see many cells in this image here on the right-hand side. The dark red or almost purple color is the nucleus of each cell. And then the lighter sort of reddish pink color, that is the staining of glycogen. And so you can see in these liver cells here that there is an awful lot of glycogen in liver cells, and that's just what this is trying to show you through a picture form, is some of the words I've talked about, some things I've said. Now we have a picture to it, in that liver cells have an awful lot of glycogen in them. And so this is really our storage form of glucose. So when we take in glucose in our diet, we store all that glucose as glycogen, and we store most of that in the liver. So think of the liver as like your food pantry, but it's the food pantry of glycogen. So it's a place that we save it, so that the times that we're not eating and our bodies need sugar to keep our metabolic processes going in our body and keep us alive, it can go to the liver and say, hey, let's get some sugar out of there and let's spread it out to all the other cells that need it. And we'll hand it out to the cells that need that sugar. And then when our cells don't really need much sugar, or they're not asking for it, we'll just store it over here in the liver, and then we'll deplete that. And as that glycogen gets depleted down to nothing, if the liver starts sending it out to all the different cells, eventually we eat again and we rebuild back up the pantry. And the liver cells get full of glucose molecules again, but again, we don't store them as single monosaccharides. We store them in the polymeric form, in the form of glycogen. So that's something you should take from this as well, is that we don't store sugar as monomers, we store sugar as polymers. And just to give you an idea of the number of monomers that can be stored in a glycogen molecule, at max capacity, a glycogen molecule can have about 120,000 glucose monomers. And also note that there's more than one glycogen molecule in a liver cell. There are many. So in one liver cell, you can have many 120,000 monomers, glycogen molecules in there. And this 120,000 is the maximum. It's like how much food can you put in your pantry. This is the maximum amount you can put in a glycogen molecule. So therefore, this number, as you haven't eaten in a while, this number will decrease greatly. And then when you eat a meal, it'll increase back up near 120,000 again. So as we eat and as we fast in between meals, this number changes. And since we're, in general, we eat 3 meals a day, 3 times a day, this glycogen molecule holds 100, you can think of it as holding 120,000 glucose monomers, and it depletes back down to near zero. Then we eat again, and it goes back up to 120,000. We use that energy throughout between the meals, and it dips down very low again, and then we eat again, and we fill it back up. So 3 times a day, this glycogen, the number of glucose monomers in this glycogen granule goes up and down throughout the day. Now here's a picture of a glycogen granule, just to kind of give you a mental image of it. It doesn't have 120,000 glucose monomers on it. First of all, this is in 2D, so we're only looking at a plane of it, but realize this would be in 3-dimensional. This would more look like a sphere than a flat disc, as it looks like here. In the center, there is a protein that starts it all off. So that's how a cell is dictated, how many of these glycogen molecules there are per cell, based on the number of these proteins. So the more of those you have in a cell, the more glycogen granules you can make. And so the liver cell has a lot of these proteins, and this protein kind of starts off the whole molecule. And then monomers are added to this protein as you eat a meal. So in glycogenolysis, this is the pathway where we take sugar molecules and break them off of the glycogen granules. So this is a time when we need energy. This is a time between meals. And to give you an idea, there's the size of a glucose molecule out here. So here's our glucose molecule, and you can see that when we just eat a meal, the glucose molecule attaches to this glycogen molecule on the left. When we need food, and we haven't eaten in a while, we can break off one of these glycogen or glucose molecules, and we can break it off and then use that glucose molecule to pass off to other cells. Now, looking back at a previous slide, when we were studying carbohydrates, we talked about glycogen before. And now we can look a little bit closer at it, and some of the main features are that most of the glucose molecules are attached via an alpha-1,4 linkage. And so you can see here's our alpha-1,4 linkage. We learned about these linkages when we studied disaccharides and polysaccharides. We looked at these glycosidic bonds, right? That's what they were called. And we know what the 1,4 means and the alpha-1,4 means. And so you can see here we have an alpha-1,4 linkage between these 2 monomers of glucose, between these 2, between these 2, between these 2, between these 2, between these 2. But occasionally we have a branch point, right? Because these glycogen granules, they branch. And the branch points are always alpha-1,6 linkages. And you can see that branch point. I'll change the color for us. And we can see that branch point right here, that there's an alpha-1,6 linkage. But when you look at a glycogen molecule, you can see most of the linkages are alpha-1,4, and occasionally we get a branch point at an alpha-1,6 linkage. And the branch points occur about every 8 to 12 residues in glycogen. Again, the point of having a glycogen molecule is that this is a storage form of a carbohydrate. We don't store sugars as monosaccharides, we store them as polysaccharides. Mammals do, including us. And this is quickly available storage, right? It's an immediate usage storage. So this is one of the first, so when our bodies need energy, this is where it goes to get that energy immediately, right away. If you're on a long time fast, you'll eventually run out of sugar. But on short fast, we can just use our glucose reserves as our primary source of long-term high volume of energy. It helps us maintain our blood sugar levels, and so this is the way we do. This is the source to increase or decrease sugar levels, is to either find glycogen and break off sugar molecules or glucose molecules and put them into the blood, or if we have too much sugar in the blood, like right after eating, the body takes the sugar out of the blood and stores it as glycogen in the liver. So let's look at the pathway of breaking down glycogen to sugar. So this would be occurring during a time when we haven't eaten in a while and our body needs energy, and so it's going to go, hey, I need some energy. Let's go to the pantry, let's go to glycogen, and let's break off some of those sugar molecules, and let's go ahead and use those as our energy source. Now, in glycogen metabolism, you will need to know the names of the enzymes that are working here. And so the first enzyme that is working here is glycogen phosphorylase. So let's say we wanted to break off this sugar right there. We're going to break that sugar off right there. So there's the end of one of the chains. And notice there's a lot of ends, but we picked one. And so in this, what we're going to do is glycogen phosphorylase in this reaction is going to take glycogen right there, and the N means the number of sugar molecules in the glycogen molecule itself, whether it's 120,000, 100,000, 50,000, doesn't matter what the number is, but that's what that N stands for. And in this reaction, glycogen phosphorylase, the enzyme, is going to take the substrate glycogen and a phosphate. And what it's going to do, it's going to break off a glucose molecule and put the phosphate on that glucose molecule it broke off. So we get glucose 1-phosphate when it does that. So it puts the phosphate on the first carbon of glucose. And then the glycogen molecule is now one less than it was before. So because it lost the glucose molecule in this reaction. So that's what that N-1 means. No matter what it was to begin with, now it's one less because we broke off one of the glucose molecules. This is a transferase reaction because we're breaking off a glucose molecule and we're sticking it onto another molecule. In this case, we're sticking it onto a phosphate. Now, back when we were talking about glycolysis, remember we discussed that when glucose enters the cell, the first reaction of that converts glucose into glucose 6-phosphate. And what that did was, remember, we talked about it trapping the glucose molecule in the cell because we put that phosphate on it. Much the same way, we have now made glucose 1-phosphate. And glucose 1-phosphate, just like glucose 6-phosphate, can't leave the cell. So at this point, even though we've broken off a glucose molecule, so to speak, not a true glucose molecule, because this is really glucose 1-phosphate, this molecule cannot leave the cell just yet. So we're going to have to go through a couple other reactions to get this to be glucose, because glucose can leave the cell. So for instance, in this case, if your brain needs glucose, the liver cell is going to break off a glucose molecule, put it out into the blood, and then the brain will pick it up from the blood and use it for energy. And so we got to get glucose into the blood. So glucose 1-phosphate can't be dumped into the blood just yet. So we're going to have to eventually go through a couple more reactions. Down here shows the pictorial of this. So here is our glucose molecule, or glycogen molecule right here. Notice the R, meaning there's a bunch of other stuff hanging on it, and you can see it. That's the thousands of sugar molecules that are still hanging on. So we're just looking at the terminal one, a terminal 2 phosphates, one right on the edge. And we're going to break this one off in the reaction. So glycogen phosphorylase, our enzyme, will use a phosphate molecule. And when it breaks off that last glucose molecule and adds the phosphate, there's our glucose 1-phosphate. And then this is the rest of the glycogen granule that's still remaining, and there's still a lot left. Now, there's not just one glycogen phosphorylase enzyme inside a cell. There's many. And so realize when the body needs energy and it calls on glycogen for that source of energy, realize lots of glycogen phosphorylases can start working on this glycogen. And this is denoted by all these red arrows. In fact, it could even be more than what I'm showing here. So there's lots of areas or substrates. So many glycogen phosphorylases could be working on this one glycogen granule at a time. And so it's just breaking off tons of glucose 1-phosphates at one moment. So don't think it's just happening one at a time. Yes, every enzyme works one at a time, but you could have many enzymes breaking down this glycogen granule. So very quickly, this glycogen could be broken down from glycogen to glucose. Now in the next reaction, the glucose 1-phosphate is isomerized. And that is happening down here. This enzyme is an isomerase. So that's the type of molecule it is. And remember, isomerases, all they do is they move around the atoms in the molecule. We don't add anything to the molecule. We don't take anything away from the molecule. We just move atoms around to change it a little bit. And in this case, all we're going to do is take the phosphate on the 1 carbon and move it to the 6 carbon, is all that's happening in this reaction. This is done by phosphoglucomutase. So the name kind of gives away what it does, phospho, because there's a phosphate on a glucose molecule. So it's a phosphorylated glucose molecule, and the mutase is a common term for isomerases. Not all isomerases use the word mutase in them, but when you see mutase, it's just referring to an isomerase. And so in this case, notice by moving that, we end up making the molecule. So here we can see on the right-hand side, the glycolysis pathway. And the molecule we just made, glucose 6-phosphate, is right here. And glucose 6-phosphate, once you make it by this pathway, has 2 choices. It could either go through the remaining steps of glycolysis to pyruvate. And I told you previously that muscle cells have glycogen. So muscle cells can do these couple of reactions to make glucose 6-phosphate, and what happens in muscle cells is just this, is that it converts glucose 6-phosphate through the remaining 9 steps of glycolysis to make pyruvate, and then therefore it can use this energy, it can use glucose 6-phosphate for energy for itself. So the skeletal muscle has glycogen and keeps all that sugar for itself when it needs it. So as the muscle cell needs energy, it just goes to glycogen and uses it. In the liver cell is much more sharing. It shares its sugar. So it's going to share its sugar with all the other cells in the body, like the brain and the heart and things like that. So unlike the muscle cells, which the skeletal muscle cells, they just keep the sugar for themselves, so they don't share at all. But in the liver cell, it only shares. So in the liver cells, typically, it's not going to do this pathway. What it's going to do is convert the glucose 6-phosphate to glucose and then put the glucose out into the blood for the other tissues. Now we have one problem we haven't addressed yet, and that is these branch points. Glycogen phosphorylase, which is the enzyme that breaks off the glucose molecules to make glucose 1-phosphate, works great on alpha-1,4 bonds. But eventually, when we start breaking off these sugars, we're going to start coming into contact with branch points. And remember, enzymes are specific. So glycogen phosphorylase is not very good at breaking alpha-1,6 linkages. It's only good at breaking alpha-1,4 linkages. So at branch points, we need to have different enzymes to help us out. So let's take a look at this. And the first thing I want to show you is this structure at the top here shows you the atomical or the molecular structure of it, showing all the atoms. What we're going to use is this one on the bottom, where each little hexagon represents a sugar, and you can see the connection points between them. We're just going to simplify the picture a little bit because we just need to see the bonds and how they're all connected. Now the colors are different here. Don't worry about the color differences. It's going to kind of show you where things are breaking and adding on and so forth. So it's going to show you some changes, or where different enzymes are going to come into play. So the colors don't mean anything, but notice we do see some alpha-1,6 linkages and some alpha-1,4 linkages in our basic structure here. And we talked about glycogen phosphorylase, how it can go through and break off alpha-1,4 linkages and break off glucoses to make glucose 1-phosphate. And that's what's shown here, is that when you're far away from a branch point, and we can see our branch point right here, we can go in and glycogen phosphorylase can break off this one and then break off this one, and then break off this one, and this one, and this one. And the ones here in this sort of red-purple color are all broken off. And we break off 8 of them here. And so that's why we have 8 phosphates. It says 8 reactions occurred, so for each reaction you need 1 phosphate, so you need 8 phosphates. And then, therefore, what you make is you make 8 glucose 1 phosphates. And you're left with this structure up here. And the reason why you can't break off any more is it's gotten way too close to the branch point. And the best way I can analogize this is like if you're using a pair of scissors and trying to cut a page out of a book. You can't get all the way to the spine usually, right? You're always going to leave a little bit behind when you try to cut a page out of a book because you can only get your scissors so close to where they're all attached together. All the pages are attached on the binding. And so, if you took that scissors and tried to cut a page out, there's going to be a little bit left over. That's what this is. You can think about this, where the alpha-1,6 linkages is where the book comes together, right? And these are the pages hanging out. And, the glycogen phosphorylase can only get so close to make that cut. And so at this point, the glycogen phosphorylase can't do any more because it's hindered from getting into the spot to make the cut. And it turns out if you're about 4 monomers away is when the problem starts coming in. Yes, it has the ability to break the alpha-1,4 bond. It just can't get the enzyme can't get into the spot to make the cut. And so we need a couple of other enzymes to help us here. And so this is more verbiage of what I just said. So I'm not going to stay on this slide because I just mentioned all these details. And so the first enzyme that helps us with this is a debranching, is a transferase molecule or enzyme. Notice it ends in ACE, so it's an enzyme. And what it does is it shifts 3 glucose residues at a time as a trisaccharide from one branch to the other. And so what it's going to do is it's going to take these ones shown in blue here that I'll square. It's going to take these guys and move them down to this spot. And so this is our resulting molecule then, right? We still haven't quite finished dealing with the branch point, but now we're starting to get close to getting rid of it. So that's what the first enzyme does. The second enzyme, the alpha-1,6 glucosidase, now notice a couple things in that name. First of all, it ends in ACE, so you know it's an enzyme, and alpha-1,6. Notice, remember, this bond right here was an alpha-1,6 bond. So this enzyme is going to break that bond off. And when it does so, it doesn't transfer it, but when it breaks it off, it actually makes a glucose molecule. And you can see that's shown here. So here is our branch point, our alpha-1,6 branch point. We're going to break this molecule off. And when we break that molecule off, we get glucose. Well, we know what glucose is. When we make glucose, that can leave the liver cell and go out into the bloodstream. So we've actually made the molecule that we want to use, that we want to release. So that's really quick and straightforward, right? Remember, the other sugar molecules, the other glucose that we broke off, didn't become glucose right away. They became glucose 1-phosphate, which will eventually become glucose 6-phosphate. So this one more directly makes glucose. In fact, in one reaction, this alpha-1,6 glucosidase makes a glucose molecule immediately, and immediately that can leave the cell into the bloodstream and go feed or give energy to other cells. So just to summarize a little bit here with a question, what 2 monosaccharides are directly made from glycogen, and what enzyme is used? So pause the video, take a moment and think about those, and then come on back and we'll chat about them. So what 2 monosaccharides are directly made from glycogen? Well, we make 2. We make glucose and we make glucose 1-phosphate. Right, these are our 2 choices. Most of the time, we make glucose 1-phosphate. Only when we come to a branch point do we actually make a glucose at any point. So most of the time we're making glucose 1-phosphate. And the enzyme that does that is glycogen phosphorylase. The enzyme that breaks off a sugar molecule, a monosaccharide, to make glucose immediately or directly is alpha-1,6 glycosidase is the enzyme that does that. But most of our work, and remember, is done by glycogen phosphorylase because most of our bonds are not alpha-1,6. Most of our bonds are alpha-1,4. Remember where the products go. The glucose product is obviously going to then just leave the cell as is, if it's in the liver cell, and then the glycogen phosphorylase, when it breaks off glucose 1-phosphate, it eventually becomes glucose 6-phosphate, and then at that point, there's a determination depending on the cell type whether it goes and finishes glycolysis out or whether it converts to glucose and leaves the cell. And in the liver cell, it's going to convert to glucose and leave the cell. Now, just to summarize this, you have a page here summarizing kind of everything we talked about when we convert glycogen to glucose. At least it shows some of those steps. It doesn't show all of them, but at least kind of gives you all in one slide all the pictures that you to help you put it all together. And just remember, if we started with this molecule up here at the top, remember, think about who's doing the work. Let's break this sugar off. That's glycogen phosphorylase. Glycogen phosphorylase breaks that one off. Glycogen phosphorylase, glycogen phosphorylase, glycogen phosphorylase, glycogen phosphorylase, glycogen phosphorylase, glycogen phosphorylase. And then eventually, once those are all broken off, now we need the transferase. So then we're going to break this off and break off the transferase will break that off, making this molecule down here. Then the alpha-1,6 glucosidase breaks off that one, and it becomes this molecule down here. And then who does the work? Well, then it's glycogen phosphorylase, glycogen phosphorylase, glycogen phosphorylase, again. It keeps going again until it gets close to another branch point. So look at how many times glycogen phosphorylase is working, and then occasionally the transferase shows up, and occasionally the alpha-1,6 shows up. But then the glycogen phosphorylase is taking care of the rest of it. I hinted at this on a previous slide, but what happens to glucose 6-phosphate when we make it, as we break down glycogen, is there's a couple different choices depending on the tissue type. So remember just a couple of things. One is when we're breaking down glycogen, the molecule we mostly break off is glucose 1-phosphate because glycogen phosphorylase is doing most of the work. And then we isomerize it to glucose 6-phosphate. And again, depending on the tissue type, depending on what happens here. I mentioned in muscle and brain as well, if they make their own glucose 6-phosphate, they're selfish and they're going to keep that energy for themselves. So they're going to process through glycolysis and eventually make pyruvate and then go off and do the citric acid cycle and electron transport. If the cell has oxygen, if it doesn't have oxygen, it's going to go through fermentation to lactate. The liver is a giving tissue. It's not going to use the energy for itself. It's going to provide it to other tissues. So the liver takes most of its glycogen 6-phosphate and converts it into glucose, which is then dumped into the blood and picked up by other tissues to use for energy. And we will not talk about that pathway, but if you've heard about the pentose phosphate pathway before in other courses, that would also be a pathway. But for this course, we're not going to discuss it. So in our studies of glycolysis, we talked about 3 regulated enzymes in glycolysis. And not only did we say they're regulated, we also said they're irreversible, meaning they only go one direction. So 7 of the enzymes can go either way, right? It can go backwards or forwards, so to speak. 3 of the enzymes only went one way. And one of those was hexokinase, shown down here in this box. And that hexokinase, just to remind you, was the step that took glucose and converted it into glucose 6-phosphate. The first reaction of glycolysis was this one. And remember, this trapped the glucose in the cell and got it ready for the isomerization reaction to come up. But notice the arrow here only went one direction. We don't see the arrow going this direction. And the reason we don't is because it can't happen. And so in the liver cell, remember, we want to be able to convert glucose 6-phosphate into glucose. but we can't go the reverse reaction. Well, we can't go the reverse reaction using hexokinase. We can go the reverse direction using another enzyme, an enzyme that can be regulated and an enzyme that can convert glucose 6-phosphate into glucose. And that molecule, or that enzyme, is shown here. It is glucose 6-phosphatase. And I underlined it there. And this allows for the conversion of glucose 6-phosphate into glucose. Now, this enzyme is present in the liver because the liver needs it so that it can make glucose and dump that glucose into the blood. In muscle and brain, they never need to make glucose and dump it into the blood because remember, they're selfish. They keep it for themselves. So this enzyme is not present in things in muscle and brain because they're never going to make glucose and dump that glucose into the blood. They're going to keep it for themselves. So they never need to be able to make glucose like this. And so therefore, it won't happen in those tissues. So again, depending on the function of the tissue depends on, well, which of these metabolic reactions occur. So looking down into the box on the lower right-hand corner, we can see that we can do the reverse reactions and the forward reaction, but it requires two completely different enzymes. So what we've talked about up to this point is we have talked about the pathway converting glycogen into glucose. And again, this is a pathway that occurs usually during times when we haven't eaten in a while, when we start to call on energy reserves to provide energy during times when we're between meals or we're in a starving or fasting condition. But then, of course, there's times when we eat a meal. So this pathway would not be the one we want on. What about converting? We need to be able to convert glucose. So after we eat a meal, we have a lot of sugar in our blood. The liver picks up a lot of that sugar and starts to store it away as glycogen. And so we need to talk about that pathway. And realize that one thing to note is these pathways aren't on at the same time, right? We want to be able to turn one on and turn one off. So eventually we're going to talk about that. How do we turn one on and turn one off when we don't want them both on at the same time? It doesn't make sense to break down glycogen to glucose at the same time we're trying to take glucose and convert it into glycogen. So again, only one of these pathways are on at a time, and it depends on the status of the cell. So here is that pathway. Some of the steps you're getting more and more familiar with, like this one. This is when glucose, after a meal, and comes into the liver cell. So here's our glucose from our meal. It came into the liver cell, and now the liver is going to start storing up this glucose, right? Because we just ate a meal, we're going to start storing up a lot of the glucose molecule. The first step in the storage process is actually the step that actually is the first step of glycolysis as well. And at this point, we've made glucose 6-phosphate using hexokinase. So that step right there is the first step of glycolysis as well. At this point, glucose 6-phosphate has a choice. If the cell needs the energy immediately, it would go through glycolysis. And I'll just abbreviate that as glyc. But it'll go through glycolysis. Well, but right after a meal, you probably have way more sugar than what your body could use at that one moment. And so therefore, most of the glucose 6-phosphate will then be, through a couple of reactions, through 3 reactions, will eventually be attached to glycogen to increase the size of the glycogen molecule. So again, we're adding one glucose at a time. Notice in the second reaction of this process uses phosphoglucomutase. Oh, that sounds familiar. Yeah, it does. It's the one that was used in the breakdown as well. It's the exact same enzyme. And so depending on whether it has more glucose 1-phosphate or glucose 6-phosphate drives the reaction. After you eat a meal, you got a ton of glucose 6-phosphate, so the reaction is going to be driven to glucose 1-phosphate. Then we have a couple of new enzymes, and eventually we get to glycogen. Now, in the breakdown of glycogen to glucose, you do need to know that pathway, the enzymes, as we talked about. In this pathway, I want you to recognize in the conversion of glucose to glycogen, you should know the first step as being part of the glycolysis pathway, and you should know that when it makes glucose 6-phosphate, there's choices to be made based on the energetics of the cell. Do I have so much energy I can just go ahead and store this, or am I low on energy in the cell and I need to process it through glycolysis? The rest of it I don't need you to know too much about, except a couple of things. One is, notice that some enzymes are used in both pathways, right? The breakdown and buildup of glycogen, the phosphoglucomutase, is used in both pathways, while some enzymes are unique. Notice glycogen phosphorylase is only part of the breakdown pathway, and glycogen synthase is only part of the glycogen pathway. And so we see this all the time in reactions that look like that the pathways are the reverse of each other. That we see a lot of common enzymes in both pathways, but then we see unique enzymes. And it's typically the unique enzymes are the ones that are regulated. Because for instance, if you turned off phosphoglucomutase, if you turn that off, it's going to turn off both pathways because both pathways need it. If you turn that enzyme on, well, then both pathways are on. And so that's a terrible enzyme to use to regulate the pathways. What's better to use to regulate pathways are the unique ones. If I turn glycogen phosphorylase off, it may shut down the breakdown pathway, but it won't necessarily shut down the build-up pathway, the glucose to glycogen pathway. So this allows us to alternatively regulate the pathways because these are unique enzymes. Said another way, that glucose to glycogen pathway is not the reverse of the glycogen to glucose pathway. It's not the perfect reverse. It's a different pathway, even though it seems like they should just be the reverse of each other. Yes, the overall consequence is the reverse, but it takes a very different pathway to do it. As we've seen several times with regulation of pathways or regulation of enzymes, we can regulate them in several ways. And some of the common ways we've seen regulation of enzymes is through covalent modification, allosteric effectors. And so in this pathway, it's no different, that these are some common ways in which we regulate this pathway as well. We're going to just look at a covalent modification that regulates this pathway. To do that, let's first understand how the cell senses, or how the cell knows whether we are low on sugar or high or have a lot of sugar in the blood. And so really, remember this, for instance, like the liver cells are just sensing or need to know if there's a lot of sugar in the blood, it's going to take it in and store it. If there's very little sugar in the cell, it's going to break down glycogen and release it to the blood. And so the two hormones that tell the liver cell whether there is sugar in the blood or not is insulin and glucagon. And glucagon's the one that tells the liver cells that there is low sugar in the blood, and therefore it needs to take some of its glycogen and dump some of the sugar into the blood. And so let's take a look at that enzyme. Let's take a look at glycogen phosphorylase. If we look at glycogen phosphorylase, under the different hormones, what would you expect to happen? So take a moment and think about it. Would you expect glucagon to turn on glycogen phosphorylase, i.e. activate it, or turn it off, inhibit it? What about insulin? Would you expect insulin to activate glycogen phosphorylase or inhibit glycogen phosphorylase? Pause the video, think about it, and then come on back and we'll continue. So the first thing to think about with this is, what does glycogen phosphorylase do? Well, glycogen phosphorylase is the enzyme, it's the first enzyme in the pathway to break down glycogen to glucose, which is shaded out here. But it's the first one to break that. So this pathway you would want on when you want more glucose, i.e. when the blood sugar level is low. And so the hormone that tells the liver cells when the blood sugar is low is glucagon. So when glucagon's around, you want to turn this pathway on, and therefore we're going to turn glycogen phosphorylase on. So we'd want glycogen phosphorylase to be active in this case. In the case of insulin, insulin's going to say there's plenty of sugar in the blood, and therefore you don't need to make any more. So therefore, in the case of insulin, you'd want this pathway off, because you don't need to make more glucose if you already have tons of glucose in the blood. And so therefore, insulin would turn this enzyme off. So this is how you kind of think about whether this enzyme should be on or off. You got to think about what the purpose of this enzyme is and what the goal is of the enzyme. So how do they do it? How do they regulate this enzyme? Well, let's take a look. So glucagon does it by using this signal transduction pathway, the G protein coupled pathway. So you can think about this as being a liver cell. Here is the blood up here, and so glucagon is in the bloodstream because it was released to tell all the cells, hey, we're low on sugar. Glucagon binds the receptor and activates the G protein coupled pathway. And what it does is that if we look down here at the bottom where this says phosphorylase, this is the glycogen phosphorylase. This, over here on this side, is where the glycogen phosphorylase is off, and this is on. And so what this does is, this pathway turns glycogen phosphorylase from off to on. And so by doing so, what this is showing is, here is the surface of the cell, glucagon binds to the receptor, and then the signal transduction pathway is activated. And that's what the multiple arrows are for. The multiple arrows meaning there's several steps in between. It's all those steps that we learned in signal transduction, and it affects the glycogen phosphorylase enzyme. And it does so by putting a phosphate on. And we can see that phosphate that was put on there. And then what it does is it turns on the pathway or activates it. So I made the arrow larger, bolder, saying that that pathway is now turned on because we phosphorylated the enzyme. We did a post-translational modification, which changed the shape of the enzyme, which made it more active. Interestingly, not only does the pathway affect glycogen phosphorylase, it actually also affects glycogen synthase. And in fact, it phosphorylates it. It does the same thing. But in this case, glycogen synthase being phosphorylated changes its shape to actually not turn it on, but turn it off. So in this case, notice I put an X on that path, on that arrow, meaning that is off. So notice with glucagon actually did two things. It turned one enzyme off and the other enzyme on, thereby shutting down one pathway and only activating the other pathway. And just as a reminder, does phosphorylation status correlate with enzyme activity? The answer is no, because obviously both these enzymes were phosphorylated, but their activities were completely opposite. Now, just as a opposite effect, and that's insulin, insulin regulates this pathway as well, but it does so when we have high levels of sugar in the blood. So in this instance, we just ate a meal, and we're trying to get that sugar out of the blood because high blood sugar levels are bad for us. So insulin's there to pull sugar out of the blood and store it as glycogen. So in this case, we want to store glycogen, so we want to convert glucose into glycogen in this scenario. So insulin does just like glucagon, it does so through a signal transduction pathway. Although this pathway is a different pathway. This pathway is called the insulin signal transduction pathway. It's very similar in that it's a cascade of effects, but you don't have to memorize this pathway. It's very similar to the G protein coupled receptor pathway in concept, but the actors, the players, the proteins, the enzymes are different. So it does so through a different way. But much like glucagon, insulin does not enter the cell. It activates by binding a receptor, it uses a signal transduction pathway, and it ends the process through a phosphorylation events or changes in phosphorylation. Its effects make sense with what the goals are. So one thing it does is it takes up glucose, or it moves the glucose transporter to the membrane of cells, so that there's more openings for glucose to get out of the blood and into the cell. So that's one thing it does. The second couple of things it does, you could probably have guessed would have happened. It's going to do something opposite that of what glucagon does, because the goals are opposite. So it's going to, instead of inhibiting glycogen synthase as glucagon does, it's going to activate that pathway, and it's going to inhibit the glycogen phosphorylase enzyme, so there it will inhibit the breakdown of glucagon to glucose. And so, again, the effects are something you can kind of start guessing is going to happen. And these effects are all activated, all three of these effects are activated by insulin binding to the insulin receptor on the surface of the liver cells. Two common disorders of glycogen metabolism are probably ones you've heard of before. One is diabetes. And remember, diabetes, we talked about earlier in the semester, is basically an abnormally high amounts of blood sugar. So in this case, we have too much sugar in the blood, i.e. we're not pulling enough sugar out of the blood and into the liver cells. And this is the most common disease in regards to glycogen. And in this case, there's several different reasons as to why this occurs, but let's just talk about one. One is maybe the person doesn't make enough insulin. So if you don't make enough insulin, you never think about the effects of insulin on liver cells. Therefore, those effects don't happen, such as the glucose transporter doesn't make it to the surface of cells. So therefore, glucose can't get into the cells because there's no transporter at the cell surface to get glucose into the cells. It doesn't turn on the pathway of converting glucose into glycogen in the cell because we never activate the glycogen synthase. We don't shut down the pathway from glycogen to glucose. And so therefore the pathways are not turned on appropriately. So in this case, sugar never enters into the cell. So a treatment for these individuals would be, we'll give them a shot of insulin, give them some insulin. If you give them insulin, it turns on all those effects, it turns on all those pathways appropriately, and sugar exits the blood and goes into the liver cells. And so these individuals usually take insulin around the time of eating a meal. Now again, this isn't the only treatment for people with diabetes, but this is just one example. Another example is hypoglycemia. This is a case where you have low blood sugar levels, and they're typically many times caused by excess insulin. And so in these individuals, usually they make insulin just fine. They respond to insulin great. It's just that they're making too much of it. And so instead of stopping the process of bringing sugar into the cell and converting it to glycogen, in these individuals, they continually, even when the levels of sugar are appropriate, they continue to pull sugar from the blood into the cells abnormally so. And so because the insulin ligand, the insulin hormone, is bound to the receptor, activating the pathway well, and so so what it does is it keeps pulling sugar from the blood abnormally and therefore lowers the concentration of sugar inside the blood.